Hi, everyone. Welcome to today. It's third session of a summer micro workshop series. My name is Jessica Wu. I'm a professor of computer science at Harvey. In our first two micro workshop sessions, we learned more about robotics and about atmospheric and climate sciences. Future workshops in this series can be found at the 2020 Summer Research and Scholarship page. And this is all under the direction of Professor Chris Clark, Associate Dean for Research and Experiential Learning. I want to cover a small bit of logistics first. I've made each of our speakers a co-host. I ask that each participant remain on mute for the duration of this call. You can choose to turn off your video feed if you wish, especially if your internet connection is weak, but it is not necessary. If you have questions for the speakers, I ask that you use the chat window and address each speaker by name. At the end of the session, I'll moderate a live question and answer session as time allows. We're going to keep to our schedule fairly strictly, out of respect for your time and theirs. Otherwise, do be aware that I will be recording this workshop for later viewing. So today, we are fortunate to be joined by 4 professors who work across several fields of computational biology. We will be learning more about specific areas of biology, including gene regulation, single-cell genomics, regulatory networks, and neurodegeneration. On the computational side, we will explore genetic models, visualization techniques, machine learning, and of course, deep learning. First speakers are quite distinguished. Amongst them, they've received 3 NSF Career Awards and 3 Alfred P. Sloan Foundation Fellowships. Both of these are amongst the most prestigious awards for outstanding early career faculty. As some background, I had the pleasure of working with each of our guests during my PhD, as each of them completed a postdoc with Manoli Kellis in his computational biology lab at MIT and through the Broad Institute. I've included some pictures here. Here are some time from our lab outings, and I hope we can all appreciate the beauty of the Charles River and Fall Village. I'm very excited to hear about my colleagues' more recent work. I'm grateful to each of them for the time they're generously giving to us. Before we begin, I would also like to thank HMC staff members Carissa Skarsgård and Latiera Rashid for their work facilitating these workshops, and for providing a web presence for sharing information. So let's go ahead then and get started. Our first speaker is Dr. Anshul Kundaje. He is an assistant professor of genetics and computer science at Stanford University. His primary research area is large-scale computational regulatory genomics. The Kundaje lab develops statistical and machine learning methods for large-scale integrative analysis of functional genomic data to decode regulatory elements and pathways across diverse cell types and tissues, and to understand their role in cellular function and disease. Previously as a postdoc at Stanford and as a research scientist at MIT, Dr. Kundaje was the lead computation analyst of the Encode project and the Roadmap Epigenomics project. You can think of these projects as spiritual successors to the Human Genome Project. In the words of one of the scientists, the Human Genome Project provided a blueprint for life. The epigenome will tell us how this whole thing is executed. Dr. Kundaje is also a recipient of the 2019 Chen Award of Excellence from the Human Genome Organization, the 2016 NIH Director's New Innovator Award, and the 2014 Alfred P. Sloan Foundation Fellowship. Today, he'll be presenting on deep learning, the regulatory code of the human genome. Please join me in welcoming Dr. Kundaje. I'll now pass over the screen sharing to our first speaker and mute my microphone.
All right, can you see my screen?
Looks good.
Okay, thanks Jessica for the introduction, and thank you all for the opportunity to present today. So I'm going to talk to you a little bit about how we use machine learning approaches, particularly deep learning, to decipher the control system of the genome. And I know we have a pretty diverse audience, so I'm going to start with a bit of introduction to genome biology. Let me see if this works. Great. So the human genome is basically a DNA biopolymer, approximately 3 billion nucleotides or letters long, 4 types of letters, typically represented as A, C, G, and T. And so it's been pretty routine that you can now sequence human genomes pretty cheaply. My lab is primarily interested in decoding the human genome, that is, deciphering all the functional letters, words, and their syntax and grammar, and also deciphering the impact of mutations in DNA sequence on genome function. So one interesting aspect of the human body is that the body obviously contains many different organs and thousands of different cell types and tissues, as shown here. What's really interesting is in a particular individual, all the cells in the body pretty much have the exact same genome. So the same genome is read and translated differently in each of these cell types and tissues. And so when you talk about genome function, the genome really needs to be interpreted in the context of each of these cell types and tissues. So let's dive a little deeper into the anatomy of the genome. If you take the nucleus of the cell and you, let's say, unwind the DNA from the cell, there are a variety of different functional elements or pieces of DNA that have important function. The first type of element is a gene. There are about which code for proteins. There are about 20,000 or so protein coding genes in the genome. They occupy just about 1.5% of the 3 billion letters in the genome. What's really interesting is different sets of genes are turned on and off in different cell types and tissues. And there are a variety of different control elements, as shown right here, regulatory control elements, which are other little pieces of DNA. And their role is to really control the activity of these genes, turn them on and off. And if you dive into the anatomy of these control elements shown here, let's say we take one of these yellow ones. These control elements are bound by various regulatory proteins, as shown here, this black triangle protein and the other two proteins right here. And these elements are also marked by various biochemical modifications, shown here as flags, colored flags. These are often referred to as epigenomic modifications. And so there's a very complex language beyond just the DNA sequence that encodes genome function in the form of these proteins that bind DNA, as well as these molecular flags that mark regulatory DNA and genes and so forth in various activity states. Now, the cool thing is you can use large-scale DNA sequencing experiments. You can sort of modify classical DNA sequencing experiments to start profiling all of these different proteins that are bound to DNA, as well as the various biochemical modifications. And in this particular figure, what I'm showing you is a series of 6 experiments. Each row right here is a different experiment. And the output of an experiment is basically a readout across the entire genome at single nucleotide resolution. And the readout tells you the likelihood of seeing a particular biochemical marker or a protein at that particular position. So you get this really beautiful digital readout across the entire genome, characterizing these various proteins that bind DNA, as well as these various biochemical modifications. And each of these modifications and protein DNA binding maps are dynamic and different across the different cell types and tissues. So the NIH funded 2 large consortia, one of which is called Encode, the Encyclopedia of DNA Elements, and the other is called the Roadmap Epigenomics Project, where basically what they did is a consortium of scientists performed a massive number of experiments, profiling hundreds of different molecular flags, as I showed you in the previous slide, across the entire genome. So this axis is the whole genome. This axis is hundreds of different molecular flags. And this axis is hundreds of cell types and tissues. So you end up with this really massive teracube of information, basically of data, and it serves as a great data set to apply machine learning methods, such as probabilistic models, deep learning, and so forth, with the view of trying to understand how the genome encodes this information and to use that data to predict effects of mutations in genetic variants. So if you're talking about deciphering genome function, let's dive a little deeper into functional elements like regulatory elements. As I mentioned, they are bound by various proteins, as shown right here. And these proteins recognize very specific DNA words, as shown right here. And these DNA words have very interesting syntax. So different combinations of words occur together in different spacing relationships, various kinds of arrangements. And these large-scale experiments that map protein DNA contacts genome-wide provide a unique opportunity to use machine learning methods to try to decipher this DNA sequence syntax of the genome. So how do we actually do that? We can formulate this problem of trying to decipher genome syntax as a machine learning prediction task. And the way to think about it is, let's say you have a particular experiment that you did on the genome and it gave you this kind of beautiful track, as you see the green track, with various values corresponding to the likelihood of a protein binding DNA. You have these values across the whole genome. So what you can do is you can take the genome, 3 billion letters, you can bin them into little chunks, let's say of 1,000 base pairs each, 1,000 letters each. And you essentially end up with a data set where you have millions of little sequences, right? These bins across the genome. Each of these sequences has a corresponding label, which can be a continuous value coming from the experiment, or you can, for example, apply some threshold to these signals and binarize it. So in this particular case, what you're seeing is a bunch of sequences that are predicted, or sorry, not predicted, that are measured to be bound by a protein of interest, and a bunch of other sequences that are found to not be bound by the protein of interest. So your goal is to try to learn a model, a predictive model that can take DNA sequences as input and learn a mapping to these molecular labels, right, that are derived from the experiment. And the beautiful thing is, since the genome is so large, 3 billion letters, you have millions of examples to train your models on, okay, to train these predictive models. So what we do is we train neural networks. Neural networks are basically one type of machine learning algorithm. In this case, what we're using the neural network to do is take DNA sequences as inputs, as I showed you in the previous slide, and learn a predictive mapping from these sequences to these molecular profiles that are measured by the experiments, okay? So one analogy to think about is basically it's like a text-to-speech converter. In this case, the text is basically DNA sequence, and the speech is basically the DNA binding profiles coming from the experiment. And so you can train these neural networks, and what the neural networks basically do is they are very advanced pattern detectors. They learn various kinds of DNA words and their complex arrangements and help translate that into the observed profiles. So just to show you how accurate these models can be, here are a few genomic loci. Here's one locus as an example, and here's another locus. And each of these different colored plots correspond to different proteins, okay? And here you're seeing the observed profile from an experiment. And this is the predicted profile from sequence by the neural network. This is another protein you can see observed and predicted, observed and predicted. And in many cases, you cannot really make out the difference between what you see from the experiment and what the model predicts just from sequence. Okay, and these are on sequences that the model is not trained on. These are on chromosomes that are not being used to train the model. So these models can really do an amazing job mapping DNA sequence to these kinds of binding profiles. So once we have these models, we can do interesting things with them. So one of the things we can do is we can reverse engineer how the model is making these predictions. So now that we have a model that can take a DNA sequence and predict these profiles, we can take the output of the model and we can track sort of the contributions of the neurons and the patterns in the model all the way back down to the individual DNA sequences and the letters. And we can get contribution scores for each nucleotide in the sequence, telling us how important those nucleotides are to make this specific prediction at that region in the genome. And using this strategy, we can take any regulatory element in the genome, and we can now provide these really beautiful high resolution scores of the predictive importance of each base, each letter in the sequence. So you can see this sequence, which is about maybe 200 base pairs long. These specific nucleotides, these letters are really important for binding of the SOX2 protein. And these two combinations of words are really important for binding of the nanode protein, right? And so this way we can use the neural network as an agnostic oracle to highlight these predictive nucleotides and these predictive words, thereby deciphering, the predictive syntax of the genome. Now, the other nice thing we can do is, since these models are so accurate, we can use them as in silico oracles. We can basically do perturbation, large-scale perturbation experiments of the genome in silico. So what we can do is take a genomic sequence, as shown here. This sequence is bound by these two proteins. One is called OCT4, the other is called NANOG. And you can see the model makes really nice predictions. This is the predicted profile for OCT4. This is the predicted profile for NANOG. And if you interpret the model says this word is very important for OCT4 binding, and these two words are very important for NANOG binding. So now you might ask the question, what would happen if I had a mutation in this sequence? What would it do to the binding profiles? Well, we can use a model like an oracle to answer that question. So here's an example. We can take this sequence and we can mutate this particular motif. We can remove this word and then push that sequence back through the model, and the model predicts what would happen. So you can see right here, if we delete this word from the sequence, the model predicts that the OCT4 profile becomes flat and the NANOG profile becomes flat, right? So that means this word is really important for OCT4 binding and NANOG binding. We can do another experiment, again, in silico, where instead of deleting the OCT4 word, we delete the NANOG word with this one right here. So we delete this one, and you notice what happens, that the binding profile for OCT4 does not change much, but the binding profile for NANOG changes a lot, right? So this tells us that this DNA word is not so important for OCT4, but is very important for NANOG, okay? So these are really nice ways of using the models as in silico oracles to do millions of different perturbation experiments. And then the ones that look really interesting are hypotheses that you can then test in the lab. So here's an example of how we test this in the lab. We can use CRISPR genome editing experiments in real cells to validate the neural network's predictions. So here's a particular DNA word. The model predicts that this is the profile of this protein. This is what the actual experiment tells us. You can see that the predicted and observed profiles are very similar. We can then take this word and mutate. You can see I mutate these two letters in the DNA. This is done using CRISPR-Cas9 genome editing. And the model predicts that this is what would happen. The profiles would get much weaker at this spot, but the profiles would stay the same at this spot. And if you do the experiment, that's exactly what you see. You see the profile disappears at this spot and reduces a little bit at this spot. So these are nice ways of validating the predictions of the experiment, and we've done pretty large-scale versions of these. Finally, one last application. Just as we are doing kind of in silico mutations of various DNA words, in this case, as you know, there are mutations in the genome that are associated with various diseases. And we can, again, use the neural network as an oracle to predict the effects of these mutations. So, for example, at this position in the genome, in this control element, healthy individuals have a G letter, whereas patients might have a C letter. And so what we can do is take the neural network and predict which nucleotides are important. The neural network predicts that in healthy individuals, the Gs are active and important, whereas in patients where the G is mutated to a C, you can see that the importance of this DNA word disappears. And this DNA word happens to be the binding site of a protein called NF-kappa B. So these give us very nice ways of testing the effects of mutations on various kinds of molecular events, and also predicting the molecular events that are in fact disrupted by these kinds of mutations. So just to end, large-scale molecular profiling datasets, like the ones I showed you, provide a really nice avenue to decipher genome function. Machine learning models can be used to learn predictive models of DNA sequence. Neural networks can learn extremely accurate models of regulatory DNA sequence, almost indistinguishable from the experiments. They can be interpreted to decipher functional DNA letters, DNA words, and syntax. That syntax can be validated using genome editing experiments. And the same kind of models can also be used to decipher disease-associated mutations. And finally, it must be obvious that computational biology is awesome. So I'll just stop there and thank various collaborators and students who helped us do this work, various funding sources, and thank you all for listening.
So let me just go ahead then before we lose everybody to thank our speakers. I certainly enjoyed hearing a lot more about what they've been doing, especially in the time since grad school and their postdocs. Gave me a lot more perspective on all these interesting problems in computational biology, where we see a small subset here at Harvey Med. So thank you again to our first speakers. Thank you for coming to our micro workshop today. There will be more workshops in this series, so please stay tuned and look at the calendar and the website.